Hello everyone, my name is Santosh Chaudhary. Today I will be talking about Archaea, the third domain of our life, and the biology of the methane formation. In this presentation, I will explain what the archaeas are, how they are different from the bacteria, and why methanogens are important in the methane production, especially in the rumen and the environment. The archaeas are fascinating microorganisms that are often less discussed than the bacteria, but they are extremely important in the biology. They are considered the third domain of life, and some archaeal group, especially the methanogens, plays major role in the methane formation. This topic is important because it connects microbiology, animal digestion, and the environmental science. First, let us look at the broad classification of a life. All living organisms are grouped into three domains: bacteria, archaea, and eukarya. Bacteria and archaea are both prokaryotic, which means they do not have the true nucleus. Eukarya includes plants, animal, fungi, and protista, which do have a nucleus. For many years, scientists thought that all prokaryotes were basically the same. However, later studies show that archaea are fundamentally different from the bacteria. That is why archaea were placed into their own separate domain. To understand this better, we need to look at how archaea were discovered. The discovery of the archaeas as a separate domain is mainly associated with the Krall and his colleague. In the 1970s, they studied ribosomal RNA sequence, especially section S rRNA, to compare different microorganisms. Their finding showed that some organisms previously thought to be bacteria were actually very different at the molecular level. This changed biological classification and led to recognition of the archaea as a completely separate domain of life. This is This was a major breakthrough because it shows that the classification should be based not only on the appearance but also on the molecular evidence. So now let us look more closely at what archaea actually are. Archaea are single-celled prokaryotic microorganisms. Although they may look similar to the bacteria under the microscope, they have a very unique biochemical and the genetic features. They are known for living in the extreme environments, but they are also found in many ordinary habitats, such as the oceans, soils, wetlands, and the digestive system of the animals. This shows that archaea are not just rare extremophiles, but an important and a widespread group of organisms. One way to understand them better is to examine their cell structure. This slide shows the structure of the archaeal cell. Like other prokaryotes, archaea do not have a nucleus or a membrane-bound organelles. Their genetic material is found in the nucleate region, and they also contain the ribosomes, cytoplasms, and the plasma membrane. However, one of the most important difference is that archaea membrane contains ether-linked lipids, which are chemically different from those found in bacteria and eukaryotes. Their cell walls are also different because they do not contain peptidoglycan, which is common in bacteria. These features help explain why archaea are considered distinct from the bacteria. Archaea are also diverse and they can be divided into major groups. Archaea are not a single uniform group. They include several types based on their habitat and the metabolism. For example, halophils thrive in the highly salty environment. Thermophils grow at the high temperature, and the methanogens produce methane under the anaerobic condition. This group shows how adaptable archaeas are and how they can survive in many different ecological niches. Among these groups, methanogens are especially important because they have major biological and environmental significance. Before we talk more about methanogens, let us look at where the archaea are found. Archaea are famous for living in the extreme habitats. This slide shows the examples such as the hot spring of the Yellowstone National Park and the Great Salt Lake of the Utah. These environments are too hot or too salty for many organisms, but certain archaeas can thrive there. At the same time, archaeas are also found in more common environments, such as the marine systems, soils, sediments, wetlands, and the animal digestive tracts. So, a key point is that the archaea are not limited to extreme places, they are much more widespread than the people often think. Since archaea are often confused with the bacteria, let us compare them directly with the bacteria. So, archaea and the bacteria are both prokaryotes, so they share some basic features such as the absence of the nucleus. They are different in very several important ways. First, their cell membrane chemistry is different. The archaeal membrane contains ether-linked lipids, while the bacterial membrane generally contains ester-linked lipids. Secondly, the bacterial wall typically contains the peptidoglycan, but archaeal cell wall do not. The third, their molecular machinery, especially for the transcription, also differs. So even though archaea may look similar to the bacteria, they are biologically and the evolutionary distinct. The next few slides summarize this difference in a very simpler visual way. This slide highlights the main structural difference between the archaeas and the bacteria. A very important point is that the archaeal membrane are more chemically stable, which helps some archaea survive under the extreme conditions. Another key difference is the cell wall. Bacteria commonly have the peptidoglycan, while the archaea do not. This difference may seem small, but they are actually very important for the classification, physiological, and survival. There are also important functional and the biochemical differences. In addition to the structure, archaea and bacteria are also different in how they carry out cellular processes. Their enzymes, metabolic pathways, and the gene expression systems are not identical. This is the one reason why archaea are so interesting scientifically. They are prokaryotes in structure, but in some molecular features they resemble eukaryotes more than the bacteria do. This becomes even clearer when we look at the molecular difference. This slide focuses on the molecular difference, especially the RNA polymerase and the promoter conscious elements. RNA polymerase is the enzyme responsible for the transcribing DNA into RNA. In the archaea, RNA polymerase is more similar in complexity to that the eukaryotes than to that the bacteria. Promoter elements, which help initiate transcription, also differ across the three domains of life. This molecular difference provides a strong evidence that the archaea are a separate domain and not just a subgroup of the bacteria. So, when we ask what makes archaea different, the answer includes both structure and the molecular biology. At this point, we can summarize the major difference by saying that archaea differ from bacteria in membrane composition, cell wall structure, and the gene expression machinery. These differences are important because they affect how archaea survive, how they function, and how they are related evolutionary to the other group organisms Understanding this difference help us appreciate why archaea deserve recognition as their own domain of life. Now let us move to one of the most important reasons why archaea matters. In this slide, it will we will explain about the vast difference in the reproduction. Both archaea and the bacteria reproduce asexually, primarily through binary fission, but archaea are also noted for the budding and the fragmentation, and endospore formation is only happening in the bacteria. One of the most important reasons archaea matters is their role in the methane production. Certain archaeas called methanogens produce methane as a part of their metabolism. Methane is an important greenhouse gas, so methanogens are significant not only in the microbiology but also in the environmental science and the climate studies. Their activity contributes to the methane cycling in the natural ecosystem and in the agricultural systems. This means the, the archaea have a direct connection to the Earth system and the global environmental processes. A major place where this happens is in the rumen of the animals. The rumen is a fermentation chamber found in animals such as cattle, sheep, and goats, most of the ruminants. It contains a complex microbiome made up of bacteria, protozoa, fungi, and archaea. These microorganisms work together to break down the fibrous plant material that the animal cannot digest on their own. During fermentation, the bacteria produce useful compounds for the animals, but they also generate the hydrogen. If hydrogen accumulates, fermentation becomes less efficient. This is where the methanogens became important. So let us focus primarily on the methanogens. Methanogens are a special group of archaea that produce methane under the anaerobic or the oxygen-free conditions. In the rumen, one common pathway is hydrogenotropic methanogenesis, shown by this equation. CO2 plus 4H2 produces CS4, that is methane, plus 2H2O, water. In simple terms, methanogens uses carbon dioxide and hydrogen to obtain the energy, and the methane is released as the byproduct. That the methane is then mainly released by the animals through the belching, also called at the eruption in the atmosphere. Methanogens are important because they remove excess hydrogen and maintain the efficient fermentation in the rumen. However, the methane they produce is also an energy loss for the animal and a greenhouse gas in the environment. So methanogens show both the biological importance of the archaea and their environmental significance. This slide summarizes the balanced role of methanogens. One, on one hand, methanogens help the rumen system by removing the excess hydrogen. This supports the efficient microbial fermentation and helps the breakdown of the fibrous feed. On the other hand, methane production has negative consequences. It represents a loss of a feed energy that the animals would otherwise use, and when released into the atmosphere, it contributes to the greenhouse gas emission. So methanogens are biologically useful within the rumen ecosystem, but they also create the challenges for the livestock efficiency and the environmental sustainability. Let us now conclude the main message of the presentation. In conclusion, archaeas are a distinct domain of life, separate from bacteria and eukaryotes. Although they are prokaryotic, they have unique structural, biological, and molecular characteristics. Among archaea, methanogens are especially important because they connect microbiology with the animal production and the climate-related processes. In the rumen, they help maintain the fermentation by removing hydrogen, and the methane they produce is an energy loss to the animals and a greenhouse gas in the atmosphere. Therefore, studying the Archaea is important not only for understanding the diversity of the life, but also for addressing practical challenges in the agriculture and the environment. Thank you.